That was rude. Oh, that was your chair, not me. Good evening, everybody. Is this really on? Bruce? Louder? Bruce, can you mic me up a little bit more? He can't hear us. Is that better? Much better. All right. Well, thank you everybody for being here at our third annual research rally. For those of you who don't know, I am Senior Associate Dean for Research, Kate Tracy, and I don't know if the wonderful medical student is here this evening joining us, but there was a medical student earlier this week at the poster session, having met me earlier in the day, asked a colleague, is Dean Tracy the Queen of Research? And so henceforth and forevermore, I'm going to be adding that to my credentials because I just think that's fun and festive. And I think it imparts sort of the whimsy of the role. So tonight I will be coming to you as Dean Queen Tracy of Research. Before we kick off the event, I'm going to ask Dean Page to come up and just make a few welcome remarks. And then we're going to get going with the great storytelling we've got in store for the evening. So Dean Page.
Thanks so much, Kate. I love this evening. It's got kind of an open mic vibe, and we got an open bar, which is amazing. Alcohol may be coming back to the University of Vermont, by the way, under the new leadership. Just a little spoiler there. I just want to take the opportunity to say hello and express my gratitude to Dean Tracy. This concept of this week and the concept of specifically this evening was hers, and it's been tremendously successful. It's one of my favorite events of this wonderful week. I'm not saying unfortunately, because I'm very fortunate to be attending a very special wedding tomorrow. I'm leaving town early in the morning, so I'm going to miss the last couple of days. I hope you have enjoyed these first 3 days as much as I have, and we'll continue to be there for the events. We have the awards tomorrow and the Stetson lecture on Friday. So it just keeps going. Tell your friends what great quality programming we have. And I just want to take the opportunity to make note of the fact that we're all here together and we're enjoying time together. And this is voluntary. We're enjoying each other's company while we think about science and get to learn about what people are doing. And that brings us back to our core values. That brings us back to our core mission. So whatever else is going on, just listening to the news coming from a board meeting earlier, it's like, I got to get that out of my head and get back here. So while we're here, let's really enjoy and celebrate the fact that we get to be here together. We get to think about science. We get to have an alcoholic or a non-alcoholic beverage together and enjoy the evening. And again, one more round of applause for our MC and the visionary for Research Celebration Week, Dean Tracy.
So thank you, Dean Page, for the very kind remarks. And it really has been an outstanding week. Just to recap a little bit, many of you were at our research laureate lecture by none other than Dan Weiss. Dan in the back there, shout out to an amazing, amazing talk to kick off Research Celebration Week. Yesterday, we had the cool experience of the first ribbon cutting event with our new president, Dr. Trump, out at the BioLabs facility at Colchester Research Facility. And then later that day, that's when I was anointed Queen of Research, Kate. So just stay with me on this. When we had our student poster session in the afternoon, and then this is our midweek event, Research Rally, which I just absolutely love because I love science and I love professional conferences and hearing about the deep work that many of us do. But we are often very specialized in our own fields, and we often don't track the details and the nuances of the great science. And so part of the point of Rally is we're storytelling creatures. So how do we make the work that we do every day relatable to people who maybe aren't in our field and don't understand what we do? I give my research address tomorrow as part of the award ceremony. So please come see Queen Kate at her address. But part of this year's message will be around the storytelling that we need to be doing. Dean Page made reference to the noise that is happening all around us. One of the ways we can find our way through together is sharing our stories about the research that we do, about the impact that we're having. And the whole point of Research Rally is to tell each other our stories. Every single year after the event is over, I run into people who say, I've known so-and-so for 25 years. I had no idea they did this cool thing. In fact, I had somebody grab me after Dan's talk and say, I've known Dan for 30 years. I had no idea his research had progressed in this way over that period of time. And we see each other all the time. So I'm sorry, what? You didn't know. So it's such a wonderful thing to watch and to hear and to see people connect to each other's stories, to each other's science, and to the important work that we do here all day, every day as part of our mission. So without further ado, I'm going to invite Dr. Tom Delaney to the mic. And Tom is an associate professor of pediatrics, and he's going to tell us a story on training is not enough, improving suicide prevention in healthcare settings. Tom?
And I have the clicker.
Oh, yeah. Got it and turn it on. There it is.
Okay, thank you. So thank you very much for this opportunity. I want to start off by just asking, please raise your hand if you know what happens when you dial 988. Okay, that's not enough people, really. So 988, since July 1st, 2024, is the U.S. National Suicide Prevention Lifeline. And what happens if you dial that from a cell phone or landline in Vermont, you'll be connected to a crisis counselor who's specifically trained to work with people experiencing suicidality or other mental health crisis. And what's remarkable about that is, like, I think we're up to 98% of those calls being answered in state, which is probably a record in the United States because they used to be answered all over the country. And when when that person answers the phone, they know about Vermont resources, and they can actually, based on where you live, they can help you get connected services, or someone you're concerned about get connected services. I just mentioned that because, first of all, not enough people know about it. And second of all, it's like one of the many, many amazing things that's happened in Vermont in the past, I would say 15 years around suicide prevention. So 20-ish years ago, we had almost no focus on suicide prevention in the state, despite having one of the highest suicide death rates in the country. And we actually had years where we had the highest death rate in the whole Eastern US, was in Vermont. And when you look at the leadership and the engagement of people, like people with lived experience, people in the mental health professional, primary care providers, specialty providers, they've all come together and they've really made some amazing changes. I'll tell you about some of those changes. So that's me. This is why we started doing this work in earnest. In 2008, I was sitting at my desk in VCHIP and I got an email from a colleague in state government, and they were writing the state's first suicide prevention grant to the feds, to an agency called SAMHSA. And they needed a nerd, basically. They needed somebody to write the QI section and the stats plan and all that stuff. And they knew I had a PhD in psychology. So I was like, oh, yeah, I'll do it. I actually knew nothing about suicide, and I had done this PhD in psychology. I knew that suicide was an outcome. That's all I knew. I just didn't learn it. I did know from personal experience, I knew that 3 young men died at my high school while I was in high school of suicide, 2 of whom were my friends. So a little bit of a personal motivation there. This is the state's motivation. If you look at the blue line, that's the US national suicide death rate. It's people per 100,000. And that's from 2013 for 12 years to 2024. The red line is Vermont. So we are always higher than the state. And actually what this graph sort of conceals the fact that our trend was actually worse until 2019. And in 2021, our trend starts getting better relative to the US trend. But we had a serious public mental health crisis, and it was suicide, and it wasn't getting enough attention until people really stepped up and started doing things like writing grants. We'll go back to this actually. So we got the grant, which was fantastic. And we spent 3 years, we developed trainings, and we implemented youth suicide prevention trainings in pretty much every middle and high school in the state. And then we got another 3-year grant. We were able to even further expand those. We got additional funding, and that allowed us to expand these sort of training models to adults and special populations, and a little bit in primary care, a little bit in emergency departments. So trying to bring suicide prevention, what we used to call gatekeeper trainings, and now we call suicide awareness and skills trainings. The idea is anybody can be involved in suicide prevention. And one of the things we taught was SAG. SAG is, if you're concerned about someone, show you care, ask the question. The question is awkward. And the question is, are you thinking of killing yourself? Are you thinking of harming yourself? That's a hard question to ask. And then the G is get the person to help. So SAG is just one example of what we taught. I'm already down to 2 minutes and 52 minutes, 52 seconds. How did that happen? Okay, so we are going to go ahead. But the problem is, we had all this amazing training. We trained thousands of people. I rode in a car with a physician, and we hit like 8 different Vermont cities, and we just did these trainings with EMTs and other first responders and clergy, right? But training, we have no evidence that training works except for shifting people's attitudes and knowledge. We know that. We know those trainings are effective. So here's an example of some of the other things that started happening about 8 to 10 years ago in Vermont. And training, obviously, Governor's Challenge is specifically focused on veterans and their families. Fantastic effort. Comprehensive Suicide Approach Grant is a CDC grant where I'm part of that. And we're focused on specific industries. So farming and construction, if you look at the stats, are disproportionately highly impacted by suicide in Vermont and nationally. So that's a very targeted approach, trying to get a little bit upstream. Emergency departments, all 14 Vermont emergency departments have now done at least one project with Vermont Program for Quality in Healthcare, where I consult with them specifically on suicide prevention in the ED. So fantastic. And Suicide Safer Pathways to Care is all about building connections between primary care practices and their local designated agency, which, if you don't know what a DA is, it's basically our county mental health departments in Vermont are called designated agencies. It's the DA system is our public mental health system in the state. So I'm going to talk to you very briefly about zero suicide. Oh, I can't see the numbers anymore. Okay, I went to sleep. Okay. So zero suicide, I actually don't want to show you that one quite yet. Zero suicide is a package of evidence-based changes that we adopted and we started implementing with this public mental health system back in 2016. And I can't even tell you all the different things. There's like 7 categories of activities. The idea is you build a pathway from initial identification of risk all the way to treatment, referral, follow-up, and post-discharge care and contact. Here's our data, because we have the training data, but we never had actual outcome data until now. Well, last January, February anyway. This is 12-year trends. That green line is the same line from the earlier chart. That's Vermont suicide death rate. Look at the orange bars. The orange bars are the height that they represent the percent of suicide decedents who were seen in the public mental health system in the year prior to dying. What this means is when we started, the blue arrow there, year 4, that's when we started zero suicide. And look where we got in year 11. Year 12 went up a little bit. I did the stats on this, and the second 6 years are significantly lower suicide death rates than the first 6 years. So something's going on. That reduction in the second 6 years translates to 90 people who didn't die because the system has actually been making changes, not just training. Training is not enough. You have to change the EHR. You have to change the type of therapies that are being offered. You have to start new things like suicide-specific screening. You have to do safety planning. There's a whole package of things that have to happen. And the sort of punchline to this is it works in the mental health care system. We've done it. We've seen this 54% reduction in deaths in the mental health care system. We don't really have it in the healthcare system. Imagine if we could get it in the healthcare system. Imagine the scale of the change we might see. Thank you.
Another round of applause for Tom. And thank you for being the first. It's always a little intimidating to be the first to go. So thank you for that, Tom. Now we're going to invite Osama Haraz, Assistant Professor of Pharmacology, up to talk to us about from tiny signals to big changes, why blood vessels hold the keys to a healthy brain. Osama?
Hey, well, good evening, everyone. Thank you very much for the introduction. Today, I'm going to talk about kind of what we do and why we do it. So we are interested in basically brain blood flow. And to get an idea about why this is important, why this actually matters, just imagine a very active city, a busy city. It's bustling with activity all the time, 24/7 active. And all the time, I mean, there are always trucks bringing resources in, and there are garbage trucks taking the waste out. Imagine the same city, but now that what is happening is that they start becoming erratic. I mean, the supplies aren't coming, and as well as the garbage starting to accumulate. And then you start to have these jams that aren't necessarily helping the people who are living in the buildings in this city. This is basically what would happen to your brain. So in a brain that is healthy, what will happen is that you have all the buildings, the neurons in your brain that are receiving the blood supply from different regions and through blood vessels. And then during disease in a neurodegeneration, for example, what will happen is that your blood vessels aren't working necessarily as they should. So in fact, I mean, there has been huge effort over the years, and this has been a big dream for scientists, I mean, to have a map for the brain, to understand the buildings that are making the blocks of this brain. So these buildings are basically the neurons in your brain. And in fact, I mean, work from UVM, as you probably all heard about last year, I mean, has kind of managed to kind of characterize the map of the brain of a fly. And this is a step that is a major step, really, that we are starting to understand all neurons and their connections, and all of this. Effort as well has been kind of going on for the human brain, the mammalian system. It hasn't yet been solved in general, but it's a major effort that we are all trying to do in order to understand what happens during neurodegeneration and what happens in a healthy brain, and then when things start breaking down. What is missing from both of these, and this is kind of huge effort that is happening worldwide, not only in the US, is the fact that we are only looking at the buildings in the city. We are not looking at the routes that will be essential for providing all of these supplies. The supplies, as well as taking rid of the waste that is coming from this city. This is where our research here at Larner College of Medicine is going about. So the example that you're looking at here is kind of a great example by one of our talented grad students sitting here. So Asser Bidair, who is a PhD student. This is an example where we are trying to really image these tracks of blood vessels within the brain of a mouse. So this is an example that you're looking at here, that this is a mouse under anesthesia. We inject these mice intravenously with a dye so that we can look at their blood vessels. We can start kind of looking at different components. We can see veins on the left, you can see arteries on the right, and we can see capillaries that are very, very tiny. But now we have the resolution that we can actually tackle questions like this. This is important because this is kind of, again, the infrastructure, the angioarchitecture that would provide for all of these brain regions what they need. Not only we can look at this, we can also look, I mean, closer. And when you're looking closer, what you have here is we can actually start looking at the traffic. Not only the structure, because in neurodegeneration, even if the structure changes, this is not something that we can actually tackle, but what is important, the traffic of this blood that is happening in different places. What you're looking at here, you can see some black shadows that are kind of going consequently, and these are actually blood cells. These are blood cells that are passing through a capillary. And we can start to understand that some of these capillaries, for example, have very slow traffic, like the ones on the southeast here, versus one with very fast traffic, like the one that here at the top. So this is very important because we can now understand not only basically the routes that where kind of the traffic will be happening, but the traffic itself that is happening. Not only we can do this, we can also start understanding how the neurons that are actually stimulating all of these responses are sending the signals and how these blood vessels are sensing these signals. We can also see traffic jams. We can start seeing kind of when capillaries or blood vessels are stopped, they stop kind of passing the blood as they should properly. So all of this is really important. Why we do what we do, and why should I care, and why should I think you care as well? I mean, what we are doing here is not necessarily only about mapping a brain. I mean, this is a great kind of thing that we are really interested in doing. But the reason for doing this is because, I mean, the fact that our brains are functioning properly and the blood supply is working as it's supposed to is kind of a way for us to maintain our memories, to maintain our kind of livelihood in general. I mean, to be able to do everything that we are doing on a daily basis. Everyone here in the audience, I would assume, probably knows at least one person or more that have neurodegeneration, someone that you love, someone that is very close to you, someone that you have lost. I personally have multiple examples that I can talk about, but this is something that is affecting every single one of us. And by understanding how the traffic works, how blood flow happens in the brain, this is kind of the very first step, I think, towards this. Not only we can understand what happens and how things break down, but we also start trying to really clear the jams or try to improve blood flow in different brain regions so that we can achieve what is needed to be achieved. That's what keeps us busy here at UVM with a great team of students and postdocs in our lab. And with that, I thank you very much for listening. Thank you.
One more round of applause for Osama. Great. And just before our interactive moment break, I will call up Dr. Tracy Madsen, Associate Professor of Emergency Medicine, who's going to talk about Beyond the Stroke Algorithms, my take on taking care of women. Take it away.
Thank you so much for having me. I'm going to be talking with you this evening about my why. So that's why I went into research, my research journey, and why I'm still here. My aspirations to become a physician and a women's health researcher really began with strong female role models that I had both growing up and into my medical training. This is Graham Biddy. She was known as Biddy for her short stature. She lived to 110 years old, 1886 to 1995. She was the matriarch of our family, and I was really lucky to get to know her. When I was a little bit older, after she died, I started hearing stories about how she had had struggles with her reproductive health. She had had 4 children. It was the early 1900s. She decided to seek a divorce from her longtime husband in order to be able to make her own choices about whether or not to have more children. This is my maternal grandmother, Graham Jane, who is Biddy's second child. She instilled in me a lifelong passion for education and for learning. She was a librarian. She went back to school after having children to get an undergraduate degree and a master's degree. She chose to have a family and a career, which was relatively rare at that time. She was a true librarian all through her life and really into the later years, even in the few years before she died. She would keep up to date on my career. She would send me articles from the newspaper about stroke, about women's health. She was like my personal librarian, helping me with my literature searches. And finally, my mom, who's a lifelong feminist, she spent the 1970s protesting, doing women's studies in college, and then volunteering at domestic violence shelters, which I ended up doing as well. These 3 women together really inspired me to pursue higher education and also to pursue a career in which I would advance understanding of women's health. So fast forward to medical school, I decided that a career in medicine was going to be how I contribute to women's health. I chose emergency medicine, partially because it was exciting, but really because I wanted to be part of the safety net that was present when people came to us for help any time of the day. I had a problem though. I wasn't sure how I was going to fit my women's health interests into a career in emergency medicine. I had started a women's health research career in undergrad and medical school when I did work in basic science labs. But then I started interviewing for residency and got lots of blank stares from program directors. I had a few questions. Why aren't you going into OBGYN? Not that I don't love OBGYN. I have a lot of OBGYN friends, but it wasn't for me. I was going to be an emergency medicine doctor. I ended up finding the perfect match at Brown University, where they were starting a new division of sex and gender-based medicine. I spent 4 years of residency, not only learning to become a doctor, but learning how to study gender disparities across the disease spectrum: cardiovascular disease, sepsis, I learned that there are sex differences in how diseases present, the efficacy of therapeutics, the accuracy of diagnostics, outcomes. And fast forward some more, how did I end up in stroke? So as I was looking at sex and gender-based differences in various diseases, stroke came up frequently. I saw patients in the ED time and time again that had waited hours, even days, before they sought care for their stroke symptoms. And I started feeling very frustrated that I couldn't fix their symptoms. I couldn't improve their outcomes after they left my hands because they didn't get the reperfusion therapy that they needed. And I learned other really interesting things about sex and gender differences in stroke. I learned that women have non-focal symptoms like pain, headache, and confusion, in addition to the focal symptoms that they have. I learned that women face longer delays to the initial CAT scan. And I learned that women are less likely to get standard of care therapies, such as reperfusion, anticoagulation, and statins for high cholesterol. So I dug deeper. I did more clinical research, quantitative and qualitative studies looking into some of these topics. But I very quickly realized that in order to make a bigger impact on women's health at the population level, I was going to need to take a step back to preventing the stroke in the first place. So I went back to school. Consistent with my family history, with all the inspiration that I had gotten from my female role models, I went back to school, gained an advanced degree in clinical and translational research, and then ended up doing a PhD in epidemiology. My husband won't let me do any more degrees. I'm done. But that's where I landed, and the rest is history. What I've been doing for the last several years is really looking at the mechanisms by which stroke risk changes over the life course for women, and why it increases so dramatically during peri and postmenopause. We're looking at things like sex-specific risk factors, migraine, endogenous sex hormones, and using lots of cool data, genomics, metabolomics, and other high-dimensional types of data to better understand the mechanism. But it hasn't been all roses. There have been some challenges. I've had lots of opportunities, and I'm very grateful, but I've also faced challenges. So rejections. I think this is part or an expected part of an academic career, but I have to keep this in mind, and I often counsel mentees about rejection and how to get through it. My particular example that I give people, this is not the only one. I submitted a K-23 3 times before being successful on the 4th. So that's perseverance. The second area of challenge, which I'm going to highlight here, is the chronic underfunding of women's health research. For decades, women have been underrepresented in medical research. This is one of the reasons I was inspired to go into women's health research. I was lucky enough to be part of a consensus study from the National Academy of Medicine. We sat in a room and we came up with recommendations for solutions to this problem at the NIH level. So giving recommendations to the NIH about how to better fund women's health. Interestingly, this was released in October 2024, which was terrible timing. I'm concerned that these recommendations will never be implemented. And then finally, a loss of trust in science and in scientists. But the challenges make the work even more important for me. I continue to be inspired by my patients, by my colleagues, by my mentees to keep doing this work. So the work of taking care of patients, the work of writing papers, writing grants, and now getting to train the next generation of scientists to be excited about women's health. And I anticipate that these challenges are not going anywhere. They may get worse. I think that my dedication to women's health is going to continue to be tested. But the patients are still here. They're still in the ED. The women are still here. They still need my help, and I'm here for it.
Keep it going. Another round of applause. All right. So I was lucky enough in my career to be mentored by President Freeman Hrabowski at the University of Maryland, Baltimore County. And one of the things that Freeman likes to say is success is never final. So I love this event. I love the research rally, and I've got a compatriot, Liz Dorman, who I forgot to give the shout out to at the beginning. Liz, Stephanie, the medical communications team, Vicki, Joanne, Sid, a lot of people have put time and energy into making these events successful. And Liz and I in particular go through every single event thinking about, ooh, what could we have done a little bit better? What could be a little bit different the next time? So this year's tweak is we've added an interactive dimension to Research Rally. So some of you are sitting in locations that have cards that look like this near you. Be afraid, be very afraid. No, I'm just kidding. This grew out of a pandemic experience that I had. During the pandemic, we're all on lockdown, which I'm built for, quite frankly. I'm a deep introvert, and being on lockdown was my jam. But I also occasionally felt the pinch of wanting to be connected. And a friend of mine talked me into this program of self-improvement. She's like, oh, come on, do it with me. And so I paid money to receive a box each month that had a surprise in it. One month it was a glass water bottle because I was supposed to hydrate, and it was habit building was what it was. And one month it was this box of cards. And I decided that what I was going to do while I was on lockdown was I was going to use my social media community to see if I could connect more richly and drive more effective conversations than I hate you, you hate me, we don't want to be family. And so what I did for a year and a half, started with this deck, was to pull a card and to post the evening cue. So what I'm going to invite you all to do, and I'm not going to shame anybody, and I'm not going to make anybody do this that doesn't want to, but I'm going to invite you to take the card, step up to the mic, and just give a short, honest answer to the crowd, because we need to build our muscles at storytelling. And we need to get to know each other in deeper, richer ways, because that's how we're going to overcome those differences. Rick is tall. I'm not as tall. He's a guy. I'm not. We grew up in different places, but you know what? We talk about our lives. We talk about our stories. We find common ground. And every single day, I learn something about being a leader from him. And I like to think occasionally he learns a thing or two from me, from the stories. So who is brave enough in this crowd to pick up the card and come to the mic? Matt Caparizzo, come on down.
So the card I have is, if you could relive a moment in your life, which one would it be? And I don't know that I can pick one, but certainly the birth of my son was incredible, and who has a kid knows what it's like in that moment. And the second would be my wedding, because the people that were there that are already not with me today, to be able to just appreciate that again would be awesome.
Thank you, Matt Caparizzo, leading by example. Others? Rebecca Wilcox, come on down.
Nothing more than a prompt. Okay, so this one is, oh yes, I'm Rebecca Wilcox. I'm the Associate Dean for Faculty. Great to see everyone. And I'm also a GI and liver pathologist and so many other things. Who or what has contributed the most to your values? And I would say that is definitely my family. And what has really inspired me today about the storytelling and thinking about research is what a privilege it is to be at an academic institution. When my father was diagnosed with amyloidosis, rare mutation, a new drug, I could go knock on somebody's door that knew what they were talking about after I'd read the literature and discuss it with them. When my mother had Parkinson's and she started getting worse right at the time of COVID. I couldn't get out there. Guess what? A clinician here not only is an expert in Parkinson's, but was doing research in telemedicine. And she got some of the best care I ever saw in that time from Vermont, even though she lived in Illinois. When my children, who are still living, so I won't reveal their issues, but I will say they went through changes that my husband and I had to take on as something new, something different, something challenging, something we had to learn a lot about. The pediatric and every part of this place, the research that we followed that was done here helped us make decisions that were the best for our kids. And in times of trouble now with the federal, I see those same people standing up at national meetings, writing legal legislation and using the research for good. And so what I value is family, but what I also see is the privilege of being here and seeing the academic research and how it affects my family and all of yours. So thank you.
Other brave souls? I mean, I did say I wouldn't publish. Steve Goldman, come to the mic, sir. Come on down.
Hi, I'm Steve Goldman. I'm the CIO of the medical school. Mine says, describe your perfect day. So in no particular order, but I usually start my day with something with music, either listening to music or playing some music, generally quietly before everybody wakes up. In my perfect day, I would get to be active. I would get to meet somebody new. I would get to learn something new, and I would get some quality time with my wife and my kids.
Any other brave souls? Please. Yeah. Oh, and I'm going to say I met this lovely, lovely, lovely person right at the beginning. She's a student in CNHS. And I was like, come on in. Come be part of the rally.
Hi, my name is Freddie, and I'm also just so grateful to have met Kate right before this. My prompt is what sounded awesome until you tried it. And this spring, I found a pair of blades just on the side of the road, and I was like, this sounds awesome. Like, I have to try this on the bike path. This seems perfect. And I tried it, and it's really, really hard. Like, it's so difficult. And they're not good rollerblades either. So I mean, I'd still recommend trying it, but watch out. You'll be sore.
I'd like a special round of applause for Freddie because it takes a lot of stuff to stand up in front of a crowd like this and share your moment. So thank you, Freddie, for doing that. Anyone else? Very true. Now, after about a week of me doing the evening queue, one of my loose colleagues said, hey, doesn't seem quite right that you're posting the question and you're watching everybody else's responses, but you're not sharing anything, Kate. If you're going to keep doing this, we think you need to share. So in the spirit of feeling like I need to share, I pulled a card. My card says, what did you learn from the worst job you ever had? I won't tell you what it was or who it was with because it might reveal things that I should just keep quietly to myself. But what I learned from that job is that leaders set the tone. Leaders show us how to be. They show us how to be in our science. They show us how to be in our educational mission. They show us how to lead. And leaders can make or break. And we can all be leaders. That was the other thing that I learned is I didn't know it was a concept called manage up. I learned to manage up in that job. And so as we go through these amazing times together, I appreciate the way you all share. I appreciate that I absolutely get up every day and go home every night believing that I have all of you in the boat with me. And I would encourage each and every one of you to A, keep telling your stories, keep linking arms. While we should be somewhat judicious in what we poke our heads above the waterline and say and do, I would invite you to keep talking about the good and important work that you do. Because if we don't tell our stories, no one will tell them for us. And with that, we're going to go to the next half. One of the other fun, look, he's already coming up. I haven't even said his name. This is perfect, actually. This is illustrative, which is a word I learned when I went to graduate school. I always thought it was illustrative. No, it's illustrative, Kate. Anyway, one of the things I love is how excited my storytellers get. And I got this beautiful email from Dr. Previs over the weekend about how excited he was putting his talk together. So it is my pleasure to invite Dr. Previs to the mic. Mike Previs, Associate Professor of Molecular Physiology and Biophysics, is going to talk to us about gene therapy wasn't the plan, how curiosity led the way. Mike?
Hey. Thanks, Dave. Let's see if I could work this thing. Okay, so I am not going to talk about my research. I'm going to talk about my career path to get there. And so I was an undergraduate at St. Michael's College, and I was a biology major, but my interest in research really didn't begin till afterwards when I was working as environmental chemist and racing sled dogs. This is a true story. So while I was doing that, I was wondering how what I was feeding them was affecting their performance. So serendipity in 2002, serendipity brought me to UVM. I met Dwight Matthews, who's an expert in metabolism and amino acid metabolism. And despite not really being qualified for a position in his lab, he was nice enough to hire me as a technician and give me an opportunity. So in the lab, I met the love of my life. So many of you know that my wife was here for 22 years. I'm not talking about her. I'm talking about a state-of-the-art mass spectrometer. So all as I wanted to know was what this thing could do. That's all I cared about. That's my primary focus. So somehow I was a technician working in the lab. I convinced Dwight to let me get cardiac muscle biopsies from Peter Van Buren, who's a cardiologist here, and be co-mentored by Jim Vigoro, who's expert in biology and had some expertise in state-of-the-art mass spectrometry at that time. Dwight, being kind of the master entrepreneur, I'm going to say that he was, he helped me craft this position where I was both a technician and a graduate student at the same time. So this was a very unique and controversial position that I was wearing two different hats at the same time. So what I did in my PhD thesis. So we knew that the heart contains cells that were packed with proteins in this dense molecular environment. But with that state-of-the-art mass spectrometer, what I could do is I could identify features within those cells, and I could actually count how many features there were. So here's two versions of Waldo. And the other thing that I noticed was Waldo never had skis on. So throughout my PhD, people said, well, okay, Mike, you're interested in Waldo, you're interested in skis, so are you going to design a drug that mimics skis? I said, no, I don't like drugs. I'm not interested in drugs, but I did like skis. So that's kind of what kept me up all night, was why Waldo wasn't wearing skis. So everybody knows this guy, Dave Warshaw. He's big advocate of athletics here, big promoter of athletics. We've all seen the video. I'm not going to re-show it. So I would talk to Dave and say, well, why wouldn't anybody want to wear skis? I just didn't understand this. And, he kind of convinced me to do a postdoc with him in 2010. So Dave got me into the lab and he said, look, Waldo's just a cartoon. I don't know why you care about this. So he had a little sleight of hand and he said, you need to get serious. You need to focus on a real actor here like Ferris Bueller. And I'm going to tell you why, because Ferris is involved with hypertrophic cardiomyopathy, a disease that affects 1 in 200 people. And we don't even know what Ferris does under normal conditions. So come to my lab, I'll teach you how to study Ferris, and then you can go and play with your cartoons again. So I said, great. I joined his lab. I developed single molecule techniques, fluorescence-based techniques in his lab. And what we were able to do is capture Ferris in many different states. So we could take pictures of Ferris within muscle. We could find his friends, so the binding partners that he interacted with. And more importantly, we could see how he influenced their behavior. So this was, really pivotal science. And I know I'm being comical, but we had like a really great run. I think we're up to 16 co-authored publications at this point. We have multiple R01s to Dave's lab, K99, R00 to my lab, all thanks to this experience with Dave. So this ended up landing me a position here at UVM as assistant professor in 2016. And, Dave really, he did pull the sleight of hand, but now I became an expert in Ferris. And what I really wanted to know is what Ferris was doing in hypertrophic cardiomyopathy. So the thing that you have to remember is that any protein in your body comes from 2 different genes. And what we knew about Ferris is that one of those genes has mutation. And the question was that mutation leading to a concept called haploinsufficiency, where you have less protein, or was there a poison peptide? So this was a big question at the time. So I teamed up with Charlene Day, who is a cardiologist at the University of Michigan, who had a tissue bank. She's now at the University of Pennsylvania. We used my love to really address this question in that tissue. And Thomas O'Leary, who's a super talented undergraduate, kind of repurposed our methods to address this question. And in 2019, we published a paper saying that haploinsufficiency is due to HCM is due to haploinsufficiency. So this was 2019. They celebrated with non-alcoholic cider. But I stayed on the emotional roller coaster with the rest of my lab because I was assistant professor, where we pursued funding in different directions. So then in 2021, I got a really cool email from Tenaya Therapeutics, who are now collaborators and colleagues of mine. And they said, hey, Mike, your paper in 2019 gave us the evidence that we need to start a gene therapy trial for the treatment of HCM. So how this works is based on our information saying that there's a defect in the gene that led to haploinsufficiency. Tenaya generates a virus. They pack DNA into that virus, and they give it to a human. And the question is whether or not they're able to restore the content of the protein and cure the disease. So my lab is actively involved with this project. They received approval for their gene therapy in 2023. We're now part of the MyPeak-1 clinical trial, where we're the first ever lab to use protein as an endpoint for a clinical trial. We're also involved with the Ridge-1 clinical trial, which is related to Ferris's cousin, and the MyClimb natural history study, which is looking at the outcomes, the probability that children will develop HCM with one of these mutations. So I just want to leave off on some serious points. So research requires dedication. So it takes time and dedication as you hone your craft. It also requires teamwork, right? So you need a good team around you, and you need to be a good teammate and support the rest of your teammates. And the final point is that research is incremental. When we're kind of in the trenches, we could just see the step that we're working on, right? But if you build really strong steps that stand the test of time, they stack on top of one another and they can have serious impact. And that's it. Yep.
Keep it going for Mike Previs. Great talk, great talk. I should have said this sooner. Please eat, drink, be merry. You know, no need to like sit and politely wait to get up if you need a snack or something to drink. This is a very informal vibe. And now it is my pleasure to invite Dr. Booker Davis, Assistant Professor of Emergency Medicine representing tonight, right? Woohoo! Booker is going to talk about the microbiome as medicine for the injured brain. Booker?
Okay, so I'm not as good as everybody else. If I don't stick to a script, I'm going well over 7 minutes. So here, let me. All right. So as a kid, even before I knew what a scientist was, I knew that I wanted to become a doctor, but I never wanted to become the type of doctor that I was familiar with, the kind that was always handing out stickers and lollipops and Robitussin every time I saw her. I imagined some person that was going to create the therapies for the ill, and that's but as much as I thought about the sick and helping out, I never thought about the brain. I always thought of the brain as this kind of computer that was in a vault, the skull, kind of locked away and protected from all illness. And then I joined the military. That's me hanging upside down over there. And as a military nurse, I saw soldiers come home with brain injuries that we couldn't heal like we could their broken bones and their shrapnel wounds. Their lives and the lives of their families would be changed forever. And later, as a scientist, I realized something simple that was more of a reset, that the brain is not like a computer at all. It's alive, obviously, but also it's connected to the body in ways that we are just beginning to fully understand. Every year, millions suffer from a traumatic brain injury. And this can be from sports, playing sports or driving, getting in a car accident or from combat. And we can save lives in the ER or on the battlefield or in battlefield hospitals. But too often what comes after is memory loss, is personality changes, dementia-like declines. And these injured brains, this is showing that these injured brains are struggling. They're struggling against the injury. They're struggling to repair themselves. And for decades, we in medicine have tried to, because the brain is locked away in that skull, have tried to coax the cells of the skull to fix the brain, fix the injury. But maybe we've been looking in the wrong place, because here's the twist. Some of the most powerful influences on brain health aren't in the skull at all, they're in the gut. And each year, each of us has trillions of microbes. These are the types of bacteria, viruses, fungi that have survived in all of Earth's climates for billions of years. And where am I? Okay. And in us, we call them the microbiome. When we choose to feed them healthy food, they break down that healthy food and give us chemicals that we call metabolites. And these metabolites can be broken down and act like text messages to our immune system and to the neurons in the brain. And after an injury, it's important to say that these gut to brain text messages can either fan the flames of inflammation or help put the fire out. We've seen that when the gut microbiome is healthy, it produces molecules that can protect the brain tissue, preserve cognitive function, and even preserve personality after an injury, after a severe injury. But when the microbiome is disturbed, recovery collapses. I'll never forget the first time I saw this in our mouse models, and I'm just going to show one slide here that you don't have to be a trained radiologist to understand. There's a hole there, and there's a smaller hole on the other side. Basically, two groups of mice received identical major injuries. And this was resembling a rod into the brain. So it's a severe injury. And one group preserved its memory, preserved its anxiety levels, preserved its tissue, brain tissue, and the other did not. Excuse me. And the other did not. And it's important to say here that the difference, again, was not in the injury. It was in the fact that after this injury, after a severe injury, we replaced the gut microbiome in these animals, and we got better outcomes. And this moment shifted my entire scientific career focus. So we began asking the questions, could the microbiome itself be medication for the brain? Could we design treatments that don't rely on drilling into the skull or anything invasive, but rely on feeding a patient? Could we even edit the microbes of the gut for the health of the patient? And the early results in my lab say yes. We found that gut microbes, and recently in the paper that's about to be released for publication, some of their metabolites, like short-chain fatty acids, can protect white matter, can protect neuronal connections, and can keep the brain resilient, even aged brains, after a severe trauma. So I'd like you to imagine this: a pill, a probiotic, even a diet plan that's not just for gut health or to keep you looking good in a bathing suit, but to that reverses or protects an injured brain, reverses that injury or protects it in case of injury. A therapy that doesn't require surgery, again, or invasive devices, but harnesses the ecosystem that's already inside of us. And this isn't just about trauma, it's about preventing dementia, easing depression, even possibly slowing Alzheimer's disease. The microbiome could be the most powerful medicine we've never realized that we have. I began this journey because I witnessed the devastation that brain injuries can cause, but I stayed because I believe that we're on the edge of something revolutionary. The brain is not alone, it has allies. And these are tiny ones living within us that know how to survive in some of the worst conditions. And if we listen to them, they may help us heal in ways that we never thought possible. The future of brain medicine might not be found in the brain itself, but in the gut. Thank you.
Keep it going for Booker. All right, very nice, very nice. And to close us out this evening, I'd like to invite my Research Advisory Council member and Associate Professor of Pediatrics, Dr. Leslie Young, who's going to talk about fire hydrants and waterfalls. Tantalizing.
Keep you all guessing with that one.
All right. So this, similar to some of the others that have come before me, is my research journey. It all started with wanting to explore research. I had just joined faculty in 2014. I was full-time clinical working in the NICU and wanted to get a taste of research and really understand if it was something I was interested in. This was the Dixie Cup phase. Fortunately, I had opportunities to experiment and kind of understand research through the Idea States Pediatric Clinical Trials Network, which we had just joined the first cycle of. And it was an opportunity for capacity building of early stage investigators, but also for understanding and being able to conduct research in rural and underserved communities. So a great opportunity. I entered into the network and came forth with ideas, and none of them were good ideas. I thought they were, but others didn't agree. And it became this puzzle of trying to figure out how to get something to move forward in this network, this opportunity that I had before me where people wanted to invest time in who I was, but needed to come up with a way to do that. So I started thinking about rural and underserved communities, the patients I serve, what was a common link between our sites, and it was, in my mind, the opioid epidemic and babies who have been exposed to opioids in utero, a common issue in our communities in rural and underserved settings. So I decided we should do a survey. We should do a survey across our 18 states and really understand what was happening at our sites to be able to understand where the areas might be for opportunities for research. And that was met with grumbles, lots and lots of grumbles. Too many surveys. We've done enough surveys. We don't need another survey. And I'm like, but you might need this survey. So I formed a group and I was head of that group, and we began putting together the survey. We met weekly. I would come up with questions and see if people liked them, and mostly people were silent on the other end of the phone. This was before Zoom. And I remember distinctly a couple weeks into this survey generation, sitting in my car at my daughter's soccer game and asking folks if they were happy with the final product, and hearing crickets, and realizing that we weren't at the final product, and realizing that still people didn't want to do this survey, and actually offering it up and saying, hey, we have this survey. I don't need to keep moving it forward. We don't have to do it in the network. If somebody else wants to do it, they can do it. And I get this frantic text from one of my senior mentors who's on the call. He's like, don't give up the survey. I'm like, okay, we're going to do this survey. So fast forward about 2 weeks, and I'm in a special meeting at our face-to-face meeting in DC for the Idea States. And it becomes clear why this individual was so hard set on me keeping this survey. And that was because the Hoehl initiative, which hadn't been named yet, but billions of dollars, $2 billion towards the opioid epidemic and research in this field, was beginning to build. And they wanted to set up research across the spectrum of care. So from adults and pain management to neonates and the babies that I care for. But they were having trouble finding anybody doing research in this area who seemed right for this position to build this foundation. Except there was this research network and there was this group who was putting together a survey. And this survey all of a sudden had risen to this level of importance that I had not expected. And so we finished up the survey, and about a month or two later, I'm sitting at the NIH in DC, having been flown down, and have the individuals from various agencies, directors of agencies sitting in front of me saying, "So, are you up for leading this?" I'm not sure what I'm leading, but heck yeah. So enter the fire hydrant stage, where I've got my Dixie cup and I'm standing in front of this fire hydrant and water is splashing in my face and I can't breathe and I can't see, but I'm going to figure this out. So it became clear that the survey was not the final product. We did an observational study across 30 sites. We gained a lot of data. I had a lot of support in that. 2 colleagues joined me who are also early stage investigators, Dr. Lori Devlin and Stephanie Merhar. And this observational study showed a ton of variation across 30 sites. Like no one's getting good care. There's just no real care plan here. So it became clear, people got increasingly excited with this work, and it became clear that they wanted a trial. So mind you, I've never conducted a trial, but I said, heck yeah, I'm here for this. So we put together, we kind of sat all in a room, understood where the gaps were, where we needed to address and put our energies. And I started putting together a trial. And what I didn't have in research understanding, I made up for as a clinician, understanding the gaps, where the needs were, and putting things together in a way where we could really address important issues. And the trial we put together was an entire care approach, a culture shift at sites. And we put together the protocol. And mind you, meanwhile, I'm Googling what's a CRF, what's a MOP, I don't know what these things are, but we're putting them together and still at the fire hydrant stage. And we have this trial. I have a minute left. I'm going to go over. That's okay. And we put together this trial and we get ready to conduct it, and COVID happens. So we pivot, and 4 months later, we implement this study. We train over 26 sites, 5,000 nurses, 50 care settings, and how to care for these babies. And that's when it gets really exciting, because the NIH loved this. And they were like, you're our person. You're doing amazing work. You need to talk to these folks. You need to present here. And this is my waterfall stage, where I'm sitting at the bottom of the waterfall and I can't find my cup and I have no clue what's going on, but keep on going. So in the waterfall stage, there's debriefing of Congress. There's a lot of media training, and media training, if you know anything about it, is done before you talk to the media. And that is not my safe place. But I had a lot of media training by the NIH and went on a media blitz with Diana Bianchi, who's the head of the NICHD at the time. And with a viewership of over 1.5 million, our articles were translated into 48 languages. And there was so much excitement over this work. Hold on. So I have not quite reached this phase yet. We're getting ready to publish the next, the follow-up of our work. But quiet waters are coming, and I'm close enough to be able to settle myself, to reflect on my past and where I've been, to reflect on where I am, and to appreciate the lessons learned along the way. The path you envision for yourself is not always the path you should take. Say yes to the survey, say yes to things that continue to build your capacity and your interests. And along the way, you can be a good advocate through the work that you do and continue to educate others.
Keep it going for Leslie. I'm going to let you stop. And now I'm going to make you reactivate for all of our great speakers and storytellers tonight. A round of applause for all of our speakers. Woo! So I would just like to say thank you from the bottom of my heart to each and every one of our speakers who stepped up to the mic and told their stories this evening, to each and every one of you who pulled that card and came up and just off the cuff shared what your response was to the prompt, and to each and every one of you who picked up that card, looked at it and thought, not me. There's room to grow. I encourage you to practice with people you trust, practice with people you mentor, practice with people that are mentoring you. The stories are so, so, so important right now. They should probably always be important. We are story-making creatures. I'm a psychologist by training. Our brains are made to understand story. And I want to encourage all of us to use our voices in positive ways to share our stories. I'm going to tease one little bit that's going to be in my address tomorrow. Several months ago, when we were in a particularly tumultuous moment with the swirl at the federal level, a group, I think it's United for Medicine Research or something like that, put out a call for stories called My Why. And it was a national campaign, and they wanted, you know, folks from organizations to go on their website, fill out their form, and it would turn into a My Why. And a couple days ago, Christian Pulcini, who I think is duly affiliated with the Department of Pediatrics and with emergency medicine, said, hey, I'm not going to be able to be at Raleigh, and I'm really bummed about that. I'm off to Vienna to talk about my fabulous research. And that thrills me for him, but I miss having him here. He's like, I'm not sure if you know that the My Why thing is up. Some of us put in our stories, you should check it out at the link. So I go to the link, it's got a beautiful map of the US, and you can click on your state. And then it shows the people who told their stories, and it gives the little, like my pocket, it's richer than my pocket card, which is like a sentence, but folks told their stories and they got packaged into these little blurbs, and they're now up, organized by state. And because I'm a data person, I'm clicking all over the place. And what I want to know is, we've got 6 for Vermont. And I was like, are we above the mean? Are we below the mean? Are we an outlier? So I'm clicking on red and blue and close to us and far from us. And believe it or not, state of 650,000 people, we've got the most stories told by our state. Yay! And one of the initiatives we'll be launching tomorrow is our own local My Why. So Stephanie thought we should take this. Stephanie, hand up in the back to Stephanie Knifick, our Director of Medical Communications. We're going to launch our own local My Why. We've got cool postcards that you can fill out. If you fill it out as a hard copy, we'll make sure it gets into the national My Why. It will also go into our local database. We also have a red cap form. So I figured out how to make a QR code that can take you to the form. Very cool. And we want to hear your stories. We want to hear your My Why. My Why is how we begin to turn the tide a little faster on the public perception of biomedical research and its importance in the ecosystem. We do such important work. Each one of you, and the people who aren't even here with us tonight, do such important work every single day. And we make impact on people's lives every single day. So my invitation to you is to feel a little bit brave and to get a little bit outside your comfort zone. And it's okay that you didn't pick up the prompt card tonight, but think about picking up the prompt and talk to your neighbor. Tell your story. Connect with somebody at the grocery store. People randomly talk to me, and I'm not even that approachable. But have your story ready and share it with the world, because the world needs more of that positivity, and we need to help people understand why we do what we do and the value that it brings, not just to Vermont, but beyond. So thank you so much for giving me your time this evening, for embracing our storytellers. Please eat, drink, talk with each other, share bits and pieces of your souls, and enjoy the rest of the week. It's been a great week so far.